morning, everybody. My name is Adrian Marquez. Thank you for coming out. My project this summer was in cell adhesion, the application of localized forces to cells. My mentor is DCU. We're working under principal investigator, associate professor Megan Valentine for the Department of Mechanical Engineering. And our funding source was the National Science Foundation. Now, the importance of cell adhesion. Cell adhesion serves several critical processes, including cell migration, signal transmission, tissue development, and repair. Now, in the center you see two neurons, and you can see how they're attached to the tissue around them and all the different cells, and they need that in order to function properly. Now, underneath you see two long rods under neurons. They're called axons, and they use those to form neuron networks. Now, they really need to connect to each other in order to communicate, to send chemical and electrical transmissions. In Alzheimer's and other neurological diseases, these neural networks fail by axon retraction. But the way that it happens and how it happens, that's what we're trying to do research on. One way to try to investigate this is to see if and how cell adhesion strength changes in diseased cells and in healthy cells. In order to do that, we need to develop methods to apply forces to the cells. And in the bottom right image, that's a rough interpretation of what we want to do. So we're going to have a cell on an extracellular matrix and attach a magnetic particle and use a magnetic tweezer, which applies magnetic force by lowering onto it. That way we could measure the force measurements that it takes to take the bead off. So this is what we want to get to, but in order to do that, we must first create and optimize the protocols using gels, and that's what I worked on this summer. So working with the gels, I used synthetic gels in order to optimize the binding and pooling protocols of known cellular proteins. And I started off with activating a cover slip with the crosslinker in order to attach polyacrylamide gel to it. So we were able to make this polyacrylamide gel to be around 10 micrometers thick, so we could attach or make the polyacrylamide gel with the biotin BSA and integrate proteins. And then I coupled the protein or the two magnetic beads that are 4.5 micrometers in diameter with streptavidin and fibronectin proteins. Now, the streptavidin and biotin properties are well known. They really like to connect to each other, so they have like near covalent bonds. So we used those two proteins at first to standardize our protocols. And then we moved on to the fibronectin integrin proteins, which are the proteins that you will see in your cells. The graphics are not really to scale in relation to each other. The beads are really large. They could be compared to like basketball, and proteins like the size of a pinball. So then I created 4 different conditions in order to test out this binding protocol. I used beads with and without gel, beads with and without proteins, and gels with and without protein. And I would take an image using microscopy before and after a wash, a standardized wash. Then I would count the beads by highlighting them with this program called ImageJ. I will highlight the beads, and then I'll have the program counted for me. And that's how I measured the percentage that came off before and after. So before I started doing my main data collection, the main thing that I saw was that there was a high binding percentage, and that was because I was using water to position the beads. So one of the first things that I started using to collect this data was to use a buffer which has ions, and it screens out the changes in the gels of the magnetic beads. So if you can see from left to right, the first 3 graphs are my non-specific binding, which is with the lack of either protein on the bead or on the gel. And you can see the graphics at the bottom. So I was still having like a 10% non-specific binding with the lack of any protein. So I was trying to reduce that and also increase and maintain my specific binding, which is the 4th bar on the right. So one of the first things that I tried to do was to increase the incubation time. So I did an overnight incubation. And then increased my non-specific binding to something a lot higher, which would tell me that my 1-hour incubation was more favorable. So looking at these protocols, I started applying some changes, and I started moving on to my fibronectin integrin test. So I already had streptavidin coupled bead, and I used a biotin labeled fibronectin to connect it to the streptavidin because they like each other. and I coated my gel with integrin. And I started having like really high non-specific binding still. So one of the things that I started looking at was maybe reducing the concentration of beads that I was applying, and that seemed to work a little bit more favorable than what I was using before. I started having like the same amount of specific binding to the right, and I was having a little bit lower non-specific binding. So by looking at this data and the one before, started looking back at the protocols and started making some changes and see what I could do to reduce the non-specific binding. And one of the improvements that I did was to use PSA, which is a protein used for cell adhesion studies. And I used it to suspend and apply my beads. So PSA has like a stability with biological reactions, and it inhibits non-specific binding. And I also used half the previous bead concentration, and I maintained my 1 hour incubation period. One other thing that I did, I removed the streptavidin biotin from my fibronectin integrin reaction. After I did that, I started observing like near like what I wanted to see as far as my non-specific binding, which was close to 0 or relatively, except for when on the third one with protein on the gel. So once I had that done, I could move on to my pooling protocol. And I started using magnetic tweezers, which are custom-built tools that we have in the lab that we use in order to manipulate and observe force measurements on the beads. So from the current pool observations that I was doing on these beads, I could note that the streptavidin biotin had much stronger bonds, and at 406 piconewtons, the streptavidin beads would not detach and then move the 2 micrometers up. And we would observe that. You can see the image on the bottom right. I took that from the magnetic tweezer. There's a diffraction difference, and you could observe the diffraction change as the bead is getting pulled. The number 1 is the magnetic bead, and the number 2 is the reference bead. So the reference bead, it doesn't move. It's made out of polystyrene, so it's non-magnetic, so it stays put. So whenever you lower the magnet, it pulls on the magnetic bead, and there's a change in diffraction in comparison to the other one. Excuse me. So the fibronectin beads, some of them would detach at 54 piconewtons, and others would detach at 406, and that's the lowest that we could put the magnet to. And other ones would stay, and they would move up about 1 micrometer. So in summary, the job that I did this summer, I established some of the protocols for binding and pulling beads to the gels, and I noticed that BSA is really good protein for use in cell adhesion studies. And through the troubleshooting and looking at the protocols and the data, we were able to reduce the non-specific binding and increase the specific binding to where we needed it to be. So that was really good. And we also noticed that the streptavidin biotin bonds were much stronger than the ones for the fibronectin integrin, which is really good to observe. Some of the future goals, we want to continue the pooling protocols to gather more data and get more specific force measurements. And we also want to perform the binding and pooling protocols with maybe different size beads to see what the difference is. Maybe it has something to do with it. Pretty sure it does. And also want to do it on cells. And applying forces to cells will not only give us an insight in seeing like the cell adhesion strength and disease cells in regards to Alzheimer's disease and other neurological diseases, but applying forces to cells will also let us see the damage being done to the cells as it's happening, and will give us insight in traumatic, blunt force trauma injuries with insight in traumatic brain injury. That's it. Thank you.